To gain a better view into how adenovirus-based gene therapy works, we're going to look at it with a current medication that uses this technology. Called Adstiladrin, or nadofaragine for the Denovic VNCG, developed by Ferring Pharmaceuticals, was approved in 2022 by the FDA. This medication is used to treat patients with high-risk Bacillus calmette-Guerin unresponsive non-muscle invasive bladder cancer with carcinoma in situ or BCG-negative MIBC with CIS. Even though this affliction may seem too specific, around 160,000 patients globally are going to be diagnosed with this condition each year. With this information in mind, let's start with the mechanism we'll discuss today. We first begin where we zoom in to the bladder. The patient here in this graphic is experiencing NMIBC. To begin, we have the patient lie down to allow a catheter to be inserted through the urethra, where Adstiladrin is injected. Adstiladrin contains adenovirus vectors containing our target DNA we want to insert into the cells. To gain a better view, let's zoom in a little bit further. Under the microscope, we have the chimeric antigen receptors, or CAR, that will soon interact with our adenovirus. In the case of the adenovirus developed by Ferring, these vectors are specifically made to target uroepithelial cells that are enhanced by a secondary molecule called SYN3, not depicted here. Continuing onwards, if CAR is activated by the adenovirus via non-covalent binding along with SYN3, a downstream signal is then initiated that signals to the cell to perform endocytosis mediated with clathrin. Clathrin is an essential element for the adenovirus as it secretes across the endosome as it helps mediate its movement towards the nucleus. Now let's move on to the nucleus. Once the endosome that is secreted across with clathrin approaches the nucleus, the protein complex begins to break down as this exposes the endosome with the cytosol. When this occurs, factors such as lysosomes and secondary enzymes begin to break down the endosome and weaken the capsid structure. When the capsid is weakened, the natural pH of the cytosol is sufficient where it can then destroy the capsid, releasing the stored complementary DNA. This DNA contains all the elements that is required to not only place itself into the DNA, but it can also cause transgenic expression of the medication's main protein of interest called interferon alpha 2b. Interferon alpha 2b can go down the following paths. Path 1 is causing external ER stress within the cell that can then produce factors that causes the cell to perform apoptosis. The second path is mediation through factors that suppresses and reverses angiogenesis, which over time starves the cancer cells, leading to their death. The third and fourth path share the same mechanism, but how it's used depends on the state of the cancer cell and how it communicates. Beginning with the third path, a dendritic cell is activated that will then downstream activate CD8 plus T cells. On these T cells is a receptor called MHC1 that plays the role of communication. Simplifying this complex crudely comes down to whether the cancer cell can reverse or cancel the initial signal created by MHC1 when it initially binds and tells the T cell to kill the cancer cell. The cancer cell in this case is unable to cancel this signal since the second head of the MHC1 receptor that is associated with this inhibiting the signal is normally non-functioning, leading to the depiction here. Finally, we have the fourth path with natural killer cells with MHC1 that try to bind to MHC1 but is different as there is no MHC1 to begin with. Normally, this change would prevent the T cells from even detecting the cancer cell, but not for the natural killer cell. No, for the natural killer cell, there is another binding event between a different receptor, and if it can't detect any form of MHC1 communication at the same time, the NK cell then signals to the cancer cell into apoptosis.